So this is our presentation on the effects of selinexor on large B-cell lymphoma. Lymphoma is a cancer of the lymphatic system. Our lymphatic system plays a huge role in protecting us. The lymphatic system consists of lymphocytes with which have important tasks such as recognizing, attacking, and scanning for any antigens. When a lymphocyte is compromised with a mutation or a gene alternation, this turn of events results in abnormal behavior. Non-Hodgkin's lymphoma has more than 70 different subtypes. These subtypes are broken down by their cell of origin. With that being said, B cells make up 90% of most NHL subtypes, and the remaining 10% are T cells. So basically, the framework of these subtypes are based off what lymphocyte is affected and the behavior of the cancer. The two behaviors are indolent lymphoma and aggressive lymphoma. Indolent lymphoma is a slow-growing cancer, and this one's more favorable considering it is more treatable. Aggressive lymphoma is exactly as it sounds. They are deemed aggressive since the behavior of the cell is growing at a much faster rate. The most common aggressive lymphoma is diffuse large B-cell lymphoma. Oh, sorry. The biology of DLBCL is basically a mix of too much cell growth and not enough cell death. The germinal center B-like subtype and the activated B-cell subtype were both identified through gene profiling. Between these two subtypes, the oncogenic pathway for overall survival is what distinguishes these two from each other. With that, the germinal center B-cell DLBCL relies on germinal center proliferation pathways, such as BCL6, PI3K, and EZH2, while activated B cell DLBCL relies on a chronic B cell receptor and survival signaling. The slide kind of goes into more detail on the protocol of what's supposed to happen and then what goes wrong.
So, how does DLBCL operate? In our bodies, we have a gene called T53, which is responsible for making P53, which is the guardian of our genome. Normally, P53 works to repair damaged DNA during our cell division. Essentially, what happens is it pauses the cell cycle and gives the cell time to fix damaged DNA during replication. If the cell can not fix the damaged DNA, P53 will trigger cell death. This allows the genome to maintain itself. However, in DLBCL, the T53 gene can mutate, which causes P53 to become dysfunctional, meaning that these cells no longer have their safety net, causing more mutations to grow. Lymphoma cells then become immortal and resistant to apoptosis. This cancer in particular is really good at evading cell growth suppression, which causes this cancer to be faster and more aggressive.
So all the stats in the following two epidemiology slides are reported from the statistics from Sarah Cancer in the United States, and it ranges from 2018 to 2022. So the population in which who is really affected kind of leans more towards males than women being affected. Racially, the non-Hispanic white population is more affected than the other races. The top graph displays the rate of new cases per 100,000 um, with race, ethnicity, and sex. This graph kind of gives a better accounting visual of who is affected as well. Um, NHL is more frequently diagnosed among people from 65 years of age to 74. Demographically, this cancer is relatively universal. However, estimated rates from GloboCan database, the United States, Australia, and Canada was observed to have the highest incidence. Incidence rates was 18.7 per 100,000 for men and women per year. NHL is the eighth leading cause of cancer in the United States. The death rate was 4.9 per 100,000 per year. The prevalence of this cancer in 2022, 830,000 people um, were estimated to be living with non-Hodgkin's lymphoma in the United States.
So the observational study that we chose to discuss related to body size and its association as a risk factor of non-Hodgkin's lymphoma. It was a pooled analysis which used 6 prospective cohort studies, and just the graph on the right kind of goes more into depth about the 6 different cohorts and just breaks it down between their study size, but also the different types of non-Hodgkin's lymphoma cases that they had obtained. I focused primarily on the DLBCL, which is the diffuse large B-cell lymphoma, as that's what we focused on primarily in this presentation. So the cohort study used about 568,000 adults. It was through 20 years of follow-up with 1-year intervals of check-ins, and it identified 11,263 cases of non-Hodgkin's lymphoma. And this was through self-reporting and then verified using medical records and cancer registries. Participants in a baseline questionnaire reported their height, their current weight, and the weight that they were when they were either 18 or 21. And then their current weight was updated periodically through those check-ins. Additionally, the scientists adjusted for age and sex when analyzing the results. They primarily used BMI, which is body mass index, which is just weight in kilograms divided by your height in meters squared. And this was used as a comparison of the results. So the average BMI at the baseline questionnaire was 25 kilograms per meter squared. And then they analyzed the young adult BMI, the current BMI, and the usual adult BMI. So in their findings, they found that both all height and your young adult BMI and adult BMI all have an association with non-Hodgkin's lymphoma. In fact, for every 10 centimeters increase in your height, you have an 18% increased risk of developing non-Hodgkin's lymphoma. However, their biggest risk factor that they found was young adult BMI. Per every 5 kilograms per meter squared increase in a young adult BMI, your risk of developing the disease increased by 14%. And this just got worse for if you were obese as a young adult. And obese is just defined as anything above 30 BMI. If you were obese as a young adult, you had a 29% increased chance of developing non-Hodgkin's lymphoma. However, the subgroup of people that they thought were most at risk of developing this cancer are individuals who were obese in both their young adulthood and their middle to late adulthood, as they had a nearly 3 times fold likelihood and chance of developing non-Hodgkin's lymphomas. And the scientists found that the findings suggest that the excess adipose tissue from obesity can cause inflammation, and this makes it a risk factor and a driver of lymphoma. I think that this was very useful in finding prevention for the disease, as it identified a major risk factor, which can be something that can be altered through either lifestyle changes or going to a doctor and receiving medication. The intervention study that was found talks about our drug of choice, Selinexor, in patients who had previously had diffuse large B-cell lymphoma, and they had relapsed or refractory. It came back again, and they had previously tried 2 to 5 treatments, and it didn't work. It had used 267 patients total. 175 were assigned to a 60 milligram dosage, and 92 were assigned to a 100 milligram dosage. Additionally, this was a phase 2 trial, so it was all about efficacy. So the analysis of efficacy was determined based on the 60 milligram dosage group. Overall, the response rate was found to be 28%, which means that 12% of the people experienced complete response. Their tumor went away completely, and they had 23 months of progression-free survival, while the other 17% had partial response, and so their tumor either did not grow or shrunk. And additionally, their median duration of the response time was 9.3 months. So on average, Selinexor extended survival to about 9 months, which is very significant, as Originally, lymphoma is a very aggressive type of cancer, and most people only had about 6 months of survival, so it added about 3 months. In the analysis of study, roughly 1.1 in 4 patients experienced meaningful tumor shrinkage, and 1 in 8 patients experienced complete pathological response. However, for the 72% of patients who did not respond, I believe that the risk benefit is low and that the benefit does not outrank the risk, as the adverse side effects can be debilitating. Additionally, the cancer is pretty aggressive, so I think it depends a lot on the quality of life and the age and the stage at which the cancer is. The side effects, as I mentioned, are debilitating, but they are reversible, and they were treated with dosage modifications and supportive care. The chart on the right just shows the most common side effects that occurred, like thrombocytopenia, which is just the loss of red blood cells and platelets, nausea, fatigue, anemia, decreased appetite, etc. There were more side effects as well, but these were just the most common that they experienced. This was very useful as an intervention because when you compared it to the standard of care at that time, those standards of care only had a response rate of about 10%. So it had increased it by 18% and it doubled the month response duration. So it proved that it was better than the current standard of care, which is why the FDA approved it.
So, Selinexor, our targeted therapy. First off, what is a targeted therapy? It refers to a drug that targets specific molecular or cellular mechanisms that cancer cells depend on to survive. So possible targets for DLBCL is a protein called XPO1. This protein is responsible for moving tumor suppressor proteins in and out of the nucleus. So earlier, back alongside the mutation of the T53 tumor suppressor gene, more mutations accumulate, causing the overexpression of the XPO1 protein, which can further exacerbate cancer growth, causing limited functional P53 to be mislocated into the cytoplasm. So scientists designed small molecules like Selinexor to inhibit XPO1. They did this testing them on lab cells and animal models. Science showed Selinexor could be beneficial, so it moved on to clinical trials.
Now we're going to be talking about how selinexor works within the body itself. First, focusing on the molecular target, so proteins such as XPO1, which is a crucial nuclear export protein, gets binded to the selinexor, which therefore shuts down the cell's ability to transport proteins out of the nucleus, along with the movement of mRNAs from the nucleus to the cytoplasm. Within the specific drug action, the strong covalent bond between selinexor and the XPO1 protein inhibits nuclear export, which forces proteins such as P53 to stay within the nucleus. This, with P53 staying inside of the nucleus, it activates cell death pathways, and other proteins such as tumor suppressor proteins and RB proteins are also affected while staying within the nucleus, causing cell death. The overall outcome within the body is that due to the restoration of these major proteins and transcriptional factors, the survival tricks are gone, leading to apoptosis and cell cycle arrest. This causes the cell to become weaker and die off, which is what we want with cancer cells. We want them to be gone. And then this furthermore relates to on why a patient might choose selinexor is because it focuses on one cancer mechanism, whereas chemotherapy damages overall more cells within the body that don't need to be damaged. Chemotherapy can hurt healthy cells, whereas the selinexor will only focus on the lymphoma cells. The basic science behind all of this was that researchers had already known that XPO1 was the major problem. They had prior knowledge that the overexpression of XPO1 correlated with worse cancer phenotypes. They saw that the nuclear export was used as a mechanism in cancer cells that inactivated tumor suppressor genes. They knew that if they could find a drug that blocked XPO1, that it would help restore the normal controls in a cell, which is what they did and which is what selinexor does. By blocking the XPO1, it would be a strong biological rationale for a targeted inhibitor as it induced lymphoma cell death. The science gave evidence that translating selective inhibitors of nuclear export, or SINE compounds, would help with the relapse of lymphoma. The overall result and patient experience is that it is mainly used for relapsed large B-cell lymphoma, as I just said. The results are that it typically responds within weeks after starting therapy, so it's a quick working drug. The tumors will either overall stop growing completely or shrink. It is dose limiting, meaning that depending on the person, they have to take more of a higher dosage of Selinexor or a lower dose, and that shows significant efficacy, meaning that there are continuous positive results within this drug. Some patient experience is that, first of all, it is taken orally, which many patients enjoy and they find it very convenient. It's a once a week pill. They don't have to go to the doctor's office as much, so it is very convenient, very easy. You can get side effects such as nausea, lowered blood counts, and low energy levels, which are all very normal, and that it does not work on hard to treat lymphomas such as like stage 4, stage 5. But overall, many patients like this because it gives them another options to those who have already tried multiple other treatments, and this one shows positive correlation with the drug and lymphoma. And those are our references.